Welcome to Anatomy and Physiology 1 Lab Lectures. My name is Kevin J. Tokoff at University of Texas at Tyler. In this particular set of lectures, we're going to be going over a very important process which is going to be based on the division of cells in eukaryotes, which include mammals, and that's the process of mitosis. Okay? And essentially what mitosis is it's cell division, whereby in most cases, a cell is going to replicate both its DNA and it's going to replicate into a new cell that theoretically is going to be identical to the cell from which it replicated. Okay, so the idea is basically I have some cell, and there's a bunch of organelles in here, and then what's going to happen is it's going to divide. So number one, you'll get essentially the same cell back, right? And then you should get out another cell that's going to be essentially identical. It's going to have similar number of organelles, number and types of organelles, and it should theoretically have the exact same DNA, barring some mutation. Okay, so notice what happened is we started essentially with one cell, and at the end of one mitotic division, we actually have two cells which theoretically should be identical unless you had some mutation. Okay, and this is the process by which cells replicate. We're also going to, in other videos, talk about a process called apoptosis. Apoptosis is going to be your extra credit assignment. So if you want the extra credit on the exam, you have to watch these videos. So this is extra credit. The idea behind apoptosis is that some cells have to be able to die. We normally don't talk about this in lower level classes very much. But a normal part of cellular metabolism is if there's too much mutation or you have an old cell, something that's outdated, has all sorts of oxidized species, it's in oxidative stress, sometimes you have to have the cell able to die. And essentially what apoptosis is programmed cell death. The cell will actually commit suicide to prevent mutation from being propagated into later divisions of cells, or I should say generations of cells. Okay, but we're going to look at mitosis in this particular video. Okay, so here's the main key that you need to keep in mind. Cells need to be able to divide. So you have in your body a finite number of cells. And so, for instance, if you're looking at skin cells, which we'll talk about in material for the second exam, skin cells have what we call mitotically active cells. These are cells that are able to do mitosis, which is cell division. Okay, so these cells are the ones that are able to divide. Specifically, we're talking about cells in the stratum basal, which is a layer of your epidermis. So they're able to divide. Okay, and so if you ever want to make new skin cells, you have to make them from pre-existing cells. So that's basically what mitosis is on another level. It's the synthesis of new cells from pre-existing ones. And so the idea is that in general, you can't make a cell out of nothing. It has to come from a cell that was already in existence. And it's how you basically replace old cells with new ones. And so we're going to talk about the steps that happen prior to mitosis in this video. Okay? The other process that we have that sort of counterbalances mitosis is apoptosis. And this is where the cell will purposely kill itself to prevent mutation from being propagated into later generations of cells. Because if there's one thing you should get out of biology type of classes, it's that cells don't want to mutate. There are mechanisms that we won't talk about that occur inside the nucleus of the cell where the DNA is to prevent its mutation. And if you have mutation, the cell will ultimately either try to repair itself or kill itself. Okay, so apoptosis is the mechanism by which we do that. That's the topic of your extra credit assignment for the test. And so you can watch that in a separate video. So in this video, we're going to talk about a process that happens prior to mitosis, and that's called interphase. Okay, interphase. And interphase is part of what we call the cell cycle. Okay, so interphase is constituted by three phases. Number one, we have the G phase. After you get through the G phase, you'll go to something called the S phase. And then after you go through the G phase and the S phase, I should say G1 phase and S phase, you get to something called the G2 phase. And once you get past this, once you get past that point, only then can you proceed into the M phase, where M stands for mitosis. Oops, that didn't come out right. mitosis. So you have to get through some processes that occur called interphase before you can get into mitosis. And really what interphase is all about is it's about cell growth and DNA replication. And we're going to find that in the next few slides. Okay? So we have to imagine what's going to be happening when the cell divides. Okay? So if you had a cell, if you had some cell, let's say, and the cell had X number of nutrients, right? And the cell went to divide and you didn't have anything else coming into the cell, well, what would essentially happen is this cell that comes off would have X over 2 nutrients, and this one would have X over 2. So hopefully that makes a little bit of sense. If you don't have any nutrients that are coming into the cell, then all the cell has to do to divide is it just has what it already had. And so you wouldn't be getting healthy cells that come out because it would be deprived of nutrients. So what ends up having to happen before you go into mitosis is whatever cell you're talking about has to intake nutrients, or it'll intake some nutrients and then biosynthesize other nutrients. Okay, so an example of that is this molecule, which is shown right here, and by no means do you have to memorize these structures. This molecule right here is called deoxy-ATP. Deoxy-ATP ultimately is a nucleotide, okay? And specifically, it's a nucleotide that will get incorporated into DNA. So we're going to find in the S phase, before mitosis, that the DNA gets replicated. And so you have to have this molecule, particularly this part. So this part of the deoxy-ATP is specifically what you have to have. Okay? Another example would be this amino acid, which is lysine. Okay? Lysine is what we call a rate-limiting nutrient for the synthesis of elastin. So elastin is a protein that's in your skin, and it gives that sort of stretchy ability to your skin. Sorry, my pen's kind of messing up. But in any case, elastin's required for skin health, and lysine's required for elastin synthesis. So the cells that divide, if they needed an amino acid, so that's what this is an amino acid. If the cell needed amino acids, then it has to intake these for protein synthesis. So that's important. This is required for protein synthesis. We're going to find that the G1 and G2 phases of interphase do a lot of protein synthesis. And then you have this guy over here. I'll do this in green. This is what we call a ribonucleotide. And this one specifically, let's see, this one looks like UTP, okay, uridine triphosphate. Okay, this one's a ribonucleotide because notice we possess the 2' hydroxyl group. And in any case, uracil is not found in DNA. So this is certainly a ribonucleotide, and this is going to be required for the process of transcription, right? The process by which we get RNA. And so the idea is that we have these nucleotides, right? So we have to replicate the DNA, okay? And this is going to require an enzyme complex called the replosome, and we use nucleotides to make the new DNA strands. If we need to make protein, we have to first synthesize the RNA, so that's what this guy is used for. It's used for RNA synthesis, and then we use the ribosome that we talked about last time and amino acids to synthesize the protein. Okay? So hopefully everybody knows that by now and it makes sense. Okay? So let's talk about G1. Basically, G1 is all about preparation for the S phase. So S phase is where we're going to replicate the DNA. Okay? S phase is all about DNA replication. And so what you have to do for DNA replication is you have to make the enzymes needed for DNA replication. And the enzymes that are needed, some of them, not all of them, this is certainly a non-exhaustive list, are shown right here. So let me just read you some of these. DNA polymerase, DNA primase, DNA ligase, topoisomerases, and a DNA helicase. So these are some of the enzymes that are required for DNA replication. Okay? Much like in RNA synthesis or transcription, we had a transcriptosome. These enzymes right here, since you're replicating the DNA, these are going to make something called the replosome. Repli for replication. So you're going to make the replosome, and that's going to help you biosynthesize new DNA. Okay? And so all of these nutrients right here, the deoxynucleotide, the amino acid, and the ribonucleotide. All of these things are essentially rate-limiting nutrients for cell division. So you're either going to have to make them, you're going to have to biosynthesize them, or you're going to have to get some of them out from outside the cell. If you're a eukaryotic organism, you get them through the diet. Okay, so you can get pieces of these through the diet. We, as humans, cannot synthesize lysine right here, but we can certainly synthesize these nucleotides. And so we can take nutrients from around the cell and make them. You don't need to know any of that for this class. I just want to hopefully make you see that we can either get the nutrients from the diet or we can make them using biosynthesis with enzymes. Okay? But hopefully it makes sense to you that if I'm going to have a cell, I've got to intake some nutrients so I don't get X/2 nutrients in each of these cells. So I can have fully functioning cells. I want to have two cells that are basically identical to the one that they came from. Okay? And G1 phase is all about the DNA, is all about the preparation for DNA replication, and it's also about cell growth. Okay, so we need to have the cell grow, and the way we have cell grows is through protein synthesis. Okay, so if you want to have the cell grow, you have to do protein synthesis. So it's not just protein synthesis from the replisome's point of view for DNA replication. You also have to have protein synthesis for things like cytoskeletal elements, like microtubules, actin, and things like that. Those have to be synthesized for the cell to be able to divide. So all in all, G1 phase is about cell growth. So hopefully that makes sense to you. And that's going to lead us into the S phase of the cell cycle. This is still part of interphase. Here's the idea behind S phase that I want you to understand. This is the process by which the DNA is replicated. And in general, that's really all you need to know about this phase. If we go back to the previous slide, G1, we made the enzymes that are responsible for DNA replication. S phase is where we use those enzymes to actually replicate the DNA. And by the way, one piece of terminology you need to be aware of. This kind of scraggly looking strands of DNA in here, this is DNA, and it sort of looks like if you took a ball of yarn and took off the yarn from the ball in the center, that's what this would look like, or it kind of looks like spaghetti noodles. This arrangement of the DNA, where it looks kind of unorganized, although it is organized, this arrangement of DNA is called chromatin. Okay, chromatin, it sort of looks like it's very disordered. It looks like it's not very regulated, but it's actually very ordered and regulated. It doesn't look like that when you first look at it, but in fact it is. This chromatin, this is the form of DNA that gets replicated. So the chromatin is essentially going to get replicated. Okay, so hopefully that makes sense. And replication of the DNA is all about the S phase, of the interphase of the cell cycle. So hopefully that makes a little bit of sense. G2 phase is not well understood. Okay, so that's a little bit of a load off of you. I'm not likely really to ask anything about G2 phase. I'll say this because it is known that the G2 phase is all about protein synthesis, just like we saw in G1. Now in G1, part of the proteins we were making were the enzymes needed to replicate the DNA. But in G2 phase, we've already replicated the DNA, so we don't need to make those enzymes. This is all about other proteins that we need to make. But all in all, okay, these proteins are going to be necessary for mitosis, as stated later in this. For entry into mitosis, we need to make those. And we also need to make components of the cytoskeleton. All in all, it's about protein synthesis and cell growth. Okay. Now, what's interesting about G2, just a side fact, is that most cancer cells actually bypass G2, which probably means there's a lot of regulation that goes on in the G2 phase prior to entry into mitosis, but it's just not well understood. Okay, so G2 is still an active area of research, perhaps something you could go into one day, but suffice it to say it's about cell growth. Okay, so those are the three phases of interphase that we're concerned with. Now, what we're going to do in the next few slides, before we actually get into the M phase, mitosis, is we're going to talk about some of the regulation of how you get into mitosis. Okay, this figure right here and the one above it, so this one right here and this one right here, I will give you these on the exam. If I ask anything about this, I'll give you these so you don't have to memorize all this stuff. but I do want you to be able to explain it. This is explaining basically the regulation of getting into mitosis. So it's not as simple as going from G1 to S phase to G2. It's not that simple and then just going into mitosis. Okay, there's a lot of regulation because you have this DNA that's replicated in the S phase. And throughout G1, S phase, and G2, you really don't want any mutation in that DNA. Okay, the cell is willing to do whatever it takes in order to prevent mutation from the DNA. Okay, and if the DNA is mutated, the cell will prevent entry into the next phase. So there are what we call checkpoints that the cell checks to see if there's any mutation in the DNA, and if there is mutation, the process of the cell cycle is halted there, and you prevent finishing that part of the cell cycle, and then you prevent going into the next phase until the mutations are repaired. And certainly you have DNA repair enzymes that do that. So we're going to talk a little bit about the regulation of that. The regulation of it is done primarily through two types of proteins. Number one is called a cyclin-dependent protein kinase, which you are more than welcome to call a CDK. That's what I'll typically refer to them as. And then there's another type of protein called a cyclin. Okay, the actual enzyme is the CDK. Okay, those are the enzymes. They're kinases, right? The cyclins are just regulatory proteins. But notice the name cyclin-dependent protein kinase. This just means that this particular protein kinase, the CDK, will only function in the presence of cyclin. So if you take away the cyclin, the CDK doesn't work. And kinases, remember, they're phosphorylases. They transfer phosphate groups to different proteins. And ultimately what this does is when CDKs phosphorylate proteins, in general, that allows entry into the next phase of the cell cycle. Okay, so that's a really key point. If a cyclin-dependent protein kinase phosphorylates other proteins, that generally means it's allowing entry into the next phase of the cell cycle. Okay, the only way to prevent entry into the next phase is to inhibit the CDK. Hopefully that makes sense. CDKs, they phosphorylate in a cascade-like fashion. They phosphorylate other proteins, and that allows entry into the next phase of the cell cycle. If you inhibit the CDK, then it will prevent entry into the next phase of the cell cycle. So hopefully that makes a little bit of sense. And now we're going to talk about how that exactly works. Okay, so what we want to do first is talk about cyclins belonging to the D class. Okay, so cyclins belonging to the D class, those include the following: CDK2, CDK4, and CDK6. Okay, they allow progression through interphase, particularly in the G1 phase. Okay, so hopefully you can see that right here. These are responsible. So CDK, oops, that wasn't supposed to do that. Let me erase that. So CDK2, right? And then you have, here's another component of CDK2, and then you have CDK4 and CDK6. Okay, those particular CDKs facilitate movement from G1 and then ultimately into the S phase. Okay, and you can look at this and see what cyclin activates them. For example, if you're looking at CDK4, for example, which cyclins activate it? Well, there's D1, D2, and D3. Okay, so what this figure is showing is it's showing the particular CDK and which cyclin activates it. So hopefully you know how to read a figure. If I'm talking about CDK2, that means cyclin E activates CDK2. Okay, and it basically shows you which phase of the cell cycle each one controls. So for example, if I'm looking at progression through G1 phase, the ones that control that are CDK4 with cyclins D1, 2, and 3, CDK6 with cyclins D1, 2, and 3, and then CDK2 with cyclins D1, 2, and 3. Okay, so these ones in particular, these ones right here, these are allowing progression through, I wasn't supposed to do that, these allow, eraser, erase that, these allow progression through G1 phase. Okay? So basically what it means is that as long as these particular CDKs, 2, 4, and 6, as long as they're activated by cyclin and there's no inhibition on them, you'll proceed through the G1 phase. Okay? A key feature you need to keep in mind is if you get mutation to the DNA, those CDKs will be inhibited and you'll halt right there. And then you'll wait until the cell repairs the DNA. As soon as the DNA is repaired, the inhibition stops and the CDKs become activated again. That's what you need to keep in mind. When CDKs are activated, you proceed through the cell cycle into the next phase. When CDKs are inhibited, the cell cycle halts and you wait for the DNA to be repaired. Okay, so hopefully that makes a little bit of sense. Okay. Then you'll eventually get to the end of G1 phase after you finish growing the cell and you finish making the DNA synthesis enzymes, okay, like DNA polymerase. And then you want to enter the S phase. That's this phase when the DNA is actually replicated. So you go from essentially 23 molecules of DNA to 46, right? So then when you want to enter the S phase, CDK, let's do this in blue, CDK2 dimerizes the cyclin E. Let me erase that because you can't see it. Dimerizes the cyclin E, and that's what's shown right here. Let me do this. CDK2 and cyclin E. Okay, when this particular CDK2 is activated, you proceed from the G1 to the S phase. Okay, so this particular combination is just talking about the transition from G1 to S. Okay, so you could imagine what inhibits it. If you have DNA damage, then DNA damage will stimulate the inhibition of CDK2, in which case you won't proceed from the G1 to the S phase. Okay, so hopefully that makes a little bit of sense. Okay, let's go on to the next slide. Oops, that went wrong way. Okay, so once you're in the S phase, once you enter it, then you have another type of CDK that's activated. CDK2 and cyclin A. This is another cyclin CDK complex that phosphorylates and allows procession through the S phase. If at any point you get DNA mutation, CDK2 is inhibited. Okay? And you'll stop there and allow time for the DNA to be fixed, and then once it's fixed, CDK2 becomes activated again, and you'll proceed through the S phase. Okay? So hopefully that makes sense by now. And once you're through the S phase, you go into G2, and then there's another type of CDK, which is CDK1 with cyclins A and B. Okay? This allows for procession from G2 into mitosis, or the M phase. So from G2 into mitosis. Okay? So what the cell is doing when it has CDK1 and cyclin A and B activated is it's allowing procession from G2 into mitosis. Now once again, if the cell has damaged DNA, mutated DNA, what happens? CDK1 becomes inhibited. Okay? And you won't proceed from G2 into mitosis. Okay? But once the cell repairs the DNA, CDK1 becomes activated again, and you proceed from G2 into mitosis. All of these things, it's the same thing over and over again. It's just which phase or which transition is it controlling. CDK1 and cyclins A and B control movement from G2 into the M phase. So hopefully that makes a little bit of sense to you. And what this figure down here is essentially showing is that these CDKs and their cyclins, they only remain activated for a short period of time. So actually it turns out that the way you control CDKs, one way to inactivate CDKs is to destroy cyclin. So for example, look here, this is cyclin D, and you can see that it's allowing procession through the G1 phase, just like we talked about before. So how do you stop the G1 phase? Well, you basically stop it by ultimately destroying cyclin D. Okay? Cyclin E, right here. Here's cyclin E. Let me box this one. Cyclin E ultimately activates CDK2, right? And you proceed from G1 to S phase. Well, what happens when you're already in the S phase? You don't need cyclin E anymore to activate CDK2, so this gets degraded. Okay? Cyclin A. Okay, let me put a triangle around this. Cyclin A activates CDK1 and allows precession from G2 into the M phase. Right? Once you're in the M phase, you don't need CDK1 activated anymore, so you degrade that. So when you're through activating a CDK, you just degrade the cyclin. There's a whole interesting physiology behind that we don't need to go into. Suffice it to say, the cyclins are essentially ubiquitinated and they're destroyed. Okay, so hopefully that makes a little bit of sense. So they only remain active for a short period of time. After they exert their effect through phosphorylation, they are inactivated by ubiquitination. Okay, but you don't need to worry about that. And by the way, like I said on the exam, I will give you this table and this one down here. Your job is really just to be able to explain it using the essays that you are given. Okay, so hopefully that makes a little bit of sense. In the next video, we're going to do the M phase, and this isn't going to take very long. This was just a little bit of important information that you understand prior to looking at the M phase of the cell cycle. So hopefully it made a little bit of sense. See you in the next video.